Dr. Khosla, your words were very, very inspiring. And I think I'm very motivated to talk about the talk that I'm going to give right now. Dr. Singh, I think one of the things that you mentioned is one book changes lives. I think that's so important. And I put one book that changed my life about maybe 10 years ago. I was wandering through, what I was trying to do. And in the last decade or so, I read this book called What is Life by Erwin Schrödinger. And it's a very, very difficult book to read. It is about quantum physics and how quantum physics reaches biology. So then I started thinking about when I started reading this book on what is life, then I started reading other books which are related to my field, which is biology. And I started reading books like James Watson's Double Helix, Siddharth Mukherjee's The Gene, the book on mitochondria, et cetera, and then I started reading all books which relate to this what is life. So one of the common features that all of these books that I have read recently in the last decade have said that this What is Life is the book that every book refers to, which is very, very surprising. So this is my circle of life of books, and these are just the same books that I showed you that I've been reading in the last 10 years. And I think it's such a fascinating area of science. It's a new science called quantum biology. Maybe we'll talk about it some other day. But thank you, Dr. Singh, for a fantastic presentation on the importance of books. So let's start with COVID. I'm going to talk about these topics, the immunology, components of the virus, the immune response to the virus, process of vaccine development, the rationale for COVID-2, and herd immunity. So today, almost 3 months into the pandemic, I think a lot of information has been bombarded from different sources. And I think even the general public who are not directly involved in science are understanding very deep science. And so there's an explosion of science in the world today. Quickly go through the slide. The virus enters the lungs. It infects cells through the ACE2 receptor. The virus replicates, and it's a cytopathic virus, and so which means it kills the cell it grows in. It induces inflammation, which is measured by proteins like cytokines, IL-1 and IL-6. And this inflammatory process, as well as the viral infection, contributes to the pathology of the virus, which affects lungs, circulatory system, the brain, and then ultimately leads to organ failure, multi-organ failure, which ultimately relates to death. So I think now we're all familiar with this very, very rapid course of the disease. So in order to understand how to find a vaccine, we need to understand a little bit about immunology. So how many of you have thought, what is the immune system of a fly? The immune system, and the humans and flies are very different, but the flies also have an immune system. Their immune system so they also don't get infected with bacteria or fungi. They have an immune system. The immune system looks like this. They have on their cell, they have a molecule called the toll-like receptor, which when it binds to these bacteria, transmits a biochemical signal into the cell. And these intracellular molecules then activate genes. In Drosophila, for example, it will activate certain enzymes, which will then kill the bacteria. So this is the immune system inside the cell of a fly. Interestingly, that mechanism of activating the immune cells by bacteria and viruses is conserved all the way to humans. In our immune system, cells like the macrophage and the granulocytes, we also have toll receptors, which they bind to these bacteria and viruses, transmit signals into the cell, activate genes, and for us, it's genes like interferons, for example. And interferons, when they are secreted, have diverse immune functions, and they activate the immune system. So that's how the innate immune response is so important in the manifestation of this disease. And I'll come to this at the end. So it, there's a huge amount of literature. I must congratulate the scientific team and the scientific publishing teams around the world to make all articles free. And almost on the day they're published, we have access to these papers. So I think it's making knowledge really, really easily accessible. So this is one slide I wanted to talk about, just which is because it relates to the immune response, as what's happening to the immune system after the virus infects the cell. So we know that the virus infects through the ACE2 and the TMP and this co-receptor called TMPRS2. When it enters the cell, it enters this organelle called the endosome, and the endosome has what are called intracellular toll receptors. I showed you previously there were extracellular toll receptors. There are intracellular toll receptors, which then, when they detect the virus, the cell tries to eliminate the virus inside the cell, just like the primitive immune system. And it activates a whole series of enzymes shown here in these blobs, which activate the gene, induces proteins like interferon, and interferons actually can kill the virus or activate cells that can kill the virus. So that's our normal immune system. But this virus is very, very intelligent, or evolved into protecting itself. So when the virus enters the cell, it escapes this endosomic structure, and it, through its RNA, starts expressing its own viral proteins like NS4, NS4A4B, NSP15, etc. And these proteins specifically bind to the signaling domain downstream of the toll receptor and block the abilities of the cell to make interferon. So if the cell cannot make interferon initially, the immune response is not mounted. So this is one of the many mechanisms by which the virus is affecting the immune response, and we are not able to generate an immune response to the virus. So what does interferon do? I'll show you. This is a slide about the immune system overall. The virus enters the cell, a lymph node, for example, and it enters the cell through an antigen presenting cell. This interaction with the virus induces interferon secretion, and this interferon secretion is like an amplifier of the immune response. So it attracts other cell types to the lymph node. It ultimately activates CD4 cells. These CD4 cells then also secrete interferons and other cytokines, which then help B cells make neutralizing antibodies, which then can clear the virus. CD4 cells also help cytotoxic T cells be activated and kill virally infected cells, for example, cells of the lung. Okay. So the left side of the slide, which is activating of cells, is called cell-mediated immunity, and the right side is the humoral immunity, both of which are very critical in developing a vaccine. So both arms of the immune response are required to eliminate the virus. Before we go into the details of the virus, we need to understand a little bit about basic immunology, because this is important to develop a vaccine. So when there are two types of antigens, there's an intracellular antigen and there's an extracellular antigens. And our immune system handles these antigens very, very differently. So when an intracellular antigen, like the virus in this case, is expressed inside the cell of a patient, the viral RNA is made into protein, and that protein is then presented on the surface of what is called MHC molecules, major histocompatibility molecules. And this antigen is presented to CD8 cells, and the CD8 cells can then kill the virus. That is called cell-mediated immunity. And this is what a scanning electron micrograph of a CD8 T cell trying to kill a virally infected cell or a tumor cell, for example, in this situation. So this is how the T cells kill, hand-to-hand combat. T cells bind to the virally infected cell and inject lethal material, and it kills the cell. On the other hand, extracellular antigens are presented to MHC class II through a completely different pathway. And this presentation presents antigens, viral antigens, to CD4 cells, which then help B cells secrete antibody. And this is the humoral immune or the antibody immunity of the virus. And the antibodies are these structures which are Y-like structures, which have variable domains and constant domains, which are noted here, which I'm telling you these 2 points because I'll talk about these two a little bit later in the talk during the vaccine. The antibody has 2 portions. One is called the antigen binding site, FAB, and what is called the FC portion, which is the stalk of the Y, which is an important portion of how the antibody works. Don Wiley at Harvard did seminal work in understanding how MHC molecules present antigen, and is one of the most influential biologists of his generation for discovering this phenomenon. Inspirational people. So let's switch gears and talk a little. We talked about the immune system. Let's talk about the virus. The virus has many proteins. It has almost 29 different proteins, but the main proteins which are on the cell surface are spike, envelope, hemagglutinin esterase, membrane, and the nucleocapsid protein. And it has an RNA genome. This is the spike protein on the cell of the virus. It has 2 components. It has this crown and it has a stalk. Now this crown and stalk, which is the spike protein of the virus, is required for the virus to enter a cell. This stalk protein has in the middle of it a protease cleavage site, which means an enzyme inside the cell has to cleave this molecule or cut this molecule at this juncture for it to enter the cell. And the cartoon in this slide I will show you is how the virus enters the cell. So if this is a spike protein here in red and purple, that's S1 and S2 domains, it binds to the ACE2 receptor shown here in blue, then it is cleaved. And the virus fuses with the cell membrane and infects the cell. So it's a complex mechanism by which it enters the cell. Recent research has demonstrated that antibodies directed against the spike protein can block entry into the virus, into the cell. Therefore, it is very logical that if we can develop a vaccine that induces an antibody response to spike, you should be able to develop protective immunity. So far, what is the research showing us, or what is studies of the immune response to patients showing us? All patients exposed to the virus develop antibodies. These antibodies are directed to the spike protein. Antibodies to spike protein can block virus in the laboratory experiments, like I just showed you. So what is in progress in terms of the immunology of antibodies? What is the level of antibody that is protective is not yet known. We will know that over the course of time. And whether both humoral and cell-mediated immunities are required for protection. We don't know that for sure yet. Antibodies are different kinds. There are neutralizing antibodies, which when they bind to the virus, they block the virus from its ability to bind to ACE2 receptors, and therefore block the entry of the virus. There are what are called non-neutralizing antibodies, which bind to the portion of the virus, which are not involved in the entry, but they still have a function. One of the functions that they could have is they bind to the FAB portion of the antibody binds to the virus, and the FC portion of the antibody can bind to another cell, which expresses what's called the FC receptor. And if it binds to the FC receptor, it can confer another mechanism for the virus to infect cells. It can infect ACE2 negative cells through this process. These antibodies are called antibody-dependent enhancing antibodies. And one question in vaccine research is, does the vaccine induce this kind of antibodies, which could make the people who are getting vaccinated more susceptible to infection? So this is one of the risks of vaccination. The course of the immune response to COVID starts with an acute infection in which there is a high viral load, whether there is no symptoms initially. Later on, a patient starts developing symptoms, and the symptoms start reducing once the immune response, the primary immune response measured by IgM and IgG responses are elicited. I also told you that T cells are required. So T cell responses also are manifested, and they typically fall in this pattern. And both T cell responses and IgG responses are required for a protective immune response against the vaccine. So let's switch gears and see what it takes to develop a vaccine. What are the key questions? What is the nature of the protective immune response? Is it just humoral antibodies, which is neutralizing antibodies, or do you also need cytotoxic T cells? What part of the virus is required to induce this immune response? What kind of vaccine, dose, frequency is required? And finally, how much vaccine needs to be made? These are the kind of questions we have to deal with right in the beginning. So what's called the target product profile, which is a requirement document for this defining what we want from a vaccine. So the goal of the vaccine is to prevent infection and also stop replication. The indication initially will be healthcare workers like doctors and nurses and all the people who take care of COVID patients, to protect them. And then susceptible individuals who are older generation people who are more susceptible, and then contacts of people who have a likelihood of getting a contact to a COVID patient. We don't know exactly how many we'll need, but we'll surely need initially, very quickly, we'll need 10 to 100 million. We may need billions, but this is a start. I told you the measures of immune efficacy will be the antibody responses and also cytotoxic T cells. And cytotoxic T cells, remember, secrete interferon gamma, which is the cytokine that this virus is able to block. We have to know which dose it induces, how long the response lasts, and whether the product is stable. You have to make a stable product. Typically, a vaccine takes about 10 to 15 years to develop, but the world at large, through amazing collaboration that Atul talked about, we're probably going to do it in less than 2 years, at least the initial vaccines. So first, in the process, you have to define what the antigen is. And a lot of research is suggesting that the spike protein of the virus may be good to, as an antigen, to develop an antibody response that will be protective. There are many ways you can make vaccines, and you've probably heard of some of these: the RNA vaccine, DNA vaccine, protein, viral vector, inactivated, and live attenuated vaccines. Then you have to develop a process to manufacture these processes, then do animal studies, clinical trials, after which you have to submit for regulatory approval, and then scale up and distribution. So this is the process of developing vaccines. And we've been doing it in months rather than years. I understand there are more than 100 clinical trials that are underway or around the world for developing a vaccine. I will talk a little bit about the Oxford chimp adeno vaccine, primarily because I want to use one vaccine to show an example of what it takes to develop a vaccine. All the other vaccines also are progressing quite well. So in order to make a vaccine, you need to educate the immune system with a portion of the virus that will elicit a memory response, which can then protect you when the real virus comes along. So that's the purpose of the vaccine. The Oxford University team has developed a vaccine which is taking the viral gene, the spike protein of the virus, and inserting it into a adenovirus, which is the virus that causes the common cold. It's a chimpanzee version of the adenovirus. And that's called chimpanzee adenovirus vaccine. That's why ChAdOx. These scientists have previously injected almost 300 individuals, and it's shown to be safe. And this is the gene of how it is done. A gene of the adenovirus is removed and replaced with the spike protein gene. Yeah. I just mentioned quickly that the role of this vaccine, which is instead of the virus, you are injecting the vaccine. The vaccine should be able to induce both cell-mediated as well as humoral immunity, and that's why I mentioned the virus needs to be presented both by the MHC class 1 intracellular pathway as well as the extracellular pathway to activate both CD4 and CD8 T cells. Results of these studies are coming at rapid succession. And this, I'll highlight, this data came out on 13th of May, just literally last week. The first sets of data from this ChAdOx study. Rhesus monkeys were vaccinated either with the ChAdOx vaccine. There were 6 monkeys who were given the ChAdOx vaccine and 3 given a control, which is the ChAdOx, same adenovirus vector, but instead of the spike protein, it has green fluorescent protein. So it's a control vaccine. The injections were intramuscular, immune responses were measured, and then 28 days later, all the animals were challenged with a live CoV-2 virus through the intranasal route to see whether the vaccine protected the monkeys. So this is the slide which shows that vaccinated animals develop an immune response. So on the left, you can see the red dots are the monkeys who are vaccinated with the COVID vaccine, and the blue are the ones that have been vaccinated with the control vaccine. And the virus neutralizing titer is on the left y-axis. You can see all the monkeys started generating virus antibodies, and they also generated T cells, as measured by this spot forming unit assay. They also make interferon gamma. Remember, I told you the vaccine induces CD4 cells to make a lot of interferon gamma. So that's a really important component of the vaccine. Before I talk to you about the challenge, I have to explain to you the respiratory system of our body. We have an upper respiratory system and a lower respiratory system. The upper respiratory system, for example, the nasal cavity, the oral cavity, the pharynx, the larynx, all of these comprise of the upper respiratory tract. And the trachea, the deep lung, the diaphragm, all of these consist of the lower respiratory tract. Why am I telling you this? Because it's going to become relevant in the next slide. The ability of the virus to grow we get infected on the upper respiratory tract, and a sample can be taken from there, from the nose swab, to look at what happens in the upper respiratory tract. And what is called the bronchoalveolar lavage, which is a tube that is put through the mouth into the lung, and a sample can be taken deep from inside the lung to see whether the virus may be there in the lower part of the respiratory tract. So this is a busy slide. This is the virus challenge. So the monkeys that had been injected either with the vaccine or the control, which is shown in red and blue, are shown in these slides. And let me just walk you through this process. I remember I told you that the lower respiratory tract samples were taken from the BAL fluid. And you can see, in general, this is number of copies of per ml. You can see generally the blue dots are, you can measure virus in the blue dots, which means monkeys who were given the control virus. But the monkeys that were vaccinated have a very low level of virus. So clearly the vaccine is working in preventing the virus from replicating in the lower lung. Now look at what's happened in the nose swab, which is the upper respiratory tract. There seems to be virus both in the vaccinated as well as the non-vaccinated or the control vaccinated group. There are red and blue dots here. So, and you can also see the other point, which is the clearance of the virus over a period of time, right? You can see that the blue bar, which is the control animals, you can detect the clinical score, which is the pneumonia, is present for a long period of time and continues to go on. In the vaccinated group, the pneumonia goes away a little bit faster. The most important thing is that the vaccinated animals do get the infection, but it is controlled. So the vaccination prevents COVID-2 induced pneumonia. It doesn't block infection. This is also confirmed by pathology. And this is shown on the top, the control animals, and we can just look at the middle bar. You can see lungs that are infiltrated with lymphocytes, which are the dark blue dots that you see. And then those in the vaccinated monkeys, you can see it's much more normal looking lung. So the summary of the animal studies so far is that it induces both cell and humoral immunity. Most importantly, it does not induce a sterilizing immunity. It's not like it doesn't prevent entry. It enables the immune system to clear the virus a little bit faster than the control group. It prevents virus from infection and replication in the lower respiratory tract, but we maybe at this stage may be less effective in the upper respiratory tract. But more importantly, it prevents virus-induced pneumonia. Other vaccine trials also are progressing, and there's a huge amount of literature that is coming out. All the other vaccine trials also have published their data on the animal studies and observed similar kind of results, meaning you induce both cell and humoral immunity, and it appears that it is a little bit more difficult to prevent infections in the upper respiratory tract. Now, in parallel, manufacturing facilities are gearing up, and Serum Institute in India is gearing up to develop large-scale manufacturing. There are many companies who are doing this all around the world. So then finally, we come to the clinical trial and the design of the clinical trial. The goal is to induce protection from COVID-2 infection and curb the disease, to induce neutralizing antibodies and to induce cytotoxic T cells. The patients will be, or the healthy subjects will be in the range of 18 to 55 years of age. There'll be a little bit more than 1,000 patients in the trial. They'll get a particular dose of the virus. Volunteers will be blinded, and they will not know whether they received the COVID-2 vaccine or a control vaccine. In this case, they will get meningococcal vaccine. And after a predefined number of individuals are infected, the study will be unblinded. And then you'll do phase 1, phase 2, phase 3 studies, which will be have to put into a big document of almost 500,000 to 1 million pages of written material, which comprises the biological license application, which will be reviewed by regulatory authorities. Inspection of the facility will take place, and then we can move on. So now we can anticipate what the results could be. In that clinical trial, if 100 people are infected and then now you unblind the study, one possibility is that all the 100 could be in the meningococcal vaccine treated group, which means the efficacy of the vaccine is 100%. 20% of them could be in the vaccine group, and 80% could be in, 80 of them could be in the meningococcal group. That'll give you an efficacy of 80%. Similarly, 30% and 50%. So there's no difference. If the result shows of the 100 individuals, 50 are in the vaccine and 50 are in the meningococcal, that means we'll say the vaccine failed. However, there's one more condition. The vaccine group could have more people infected than the control group. That would mean the efficacy is minus 20%, which will make the vaccine dangerous. This is also a very distinct possibility that the vaccine could do because of those enhancing antibodies that the vaccine could induce. So that's why a clinical trial is very important to do before you know vaccine works. And in my final slides, I will show you that the importance of a vaccine is to generate efficacy and safety, the scientific evidence, but in addition, it has to have economic, social, and now political implications of the vaccine also. Finally, what will the vaccine do? It will induce herd immunity, where you have a group of people who are either uninfected, previously infected shown in the black dots, or currently infected shown in the red dots. And these are the sequestered people who are infected in the hospital. If you have a sufficient number of people vaccinated, then if you look at this red dot in the middle, this person can only infect this green dot and not anyone else, because everyone else around is either vaccinated or previously infected. This is the concept of herd immunity. And I'll end here by saying that I talked about the basic immunology, the components of the virus, the immune response to the virus, a rational design, and herd immunity. So I'll stop here, and thank you very much for the wonderful opportunity.